My name is Dokholyan. I'm a Michael Hooker Distinguished Professor of Biochemistry and Biophysics. I have been at UNC since 2002. I came actually from the physics background, but I got very interested in protein misfolding diseases and ALS in particular, and since 2002 basically dedicated my life finding the cure for ALS. ALS, as you know, is a motor neuron disease where motor neurons die eventually, and because of that muscles don't get exercised and they atrophy. That leads to paralysis and eventual death. We don't know exactly why motor neurons die. There are many proteins that are involved, and because of that, this is where personalized medicine is going to be extremely important, because there are really multiple diseases collected under one umbrella called ALS. We were very lucky to have Shafiq Karam coming to our campus and establishing ALS clinic. We started collaborating with him and devised several projects along those lines, and one of them is to monitor post-translational modifications in superoxide dismutase and this protein, and trying to understand whether it's present in higher amounts than in normal individuals. Second big project is we're trying to get to the roots of why this disease happens and why this protein aggregates. What we discovered is the protein has to dissociate, it has to fall apart. So that's step one. But step two, it has to assemble into trimers. So we have two-pronged strategy. One, stabilizing the dimer. The native dimer has a cavity at the bottom, let's say. And what we're trying to do is we're trying to screen small molecules computationally that would fit into this cavity inside and would bring these pieces together and not let them fall apart, even if they are modified by post-translational modifications. The second strategy is when you already have these trimers have to assemble. So you want to prevent that. So another way, because we know their structures, what we want to do is to stick a small molecule that would disrupt the trimer formation, therefore disrupting formation of toxic species. So one of the projects is identifying biomarkers or identifying signs of whether a person may develop ALS. For that, we work very closely with Shafiq, who interacts with patients, and they donate their blood samples and we extract blood, trying to find this biomarker. Another big project that we have is we're trying to alleviate the disease by targeting proteins that scavenge calcium in cells using vitamin D. And we're trying to understand whether this approach is going to be helpful for viability of motor neurons. And our preliminary results suggest that indeed, if we treat cells with calcium, they live longer. But that's cells in a dish. We are far from making definite statements. The most promising, I think, is that trying to prolong patients' life with vitamin D studies that we are doing with Shafiq. We see strong effect in living cells, and Shafiq has evidence as well that it may work in patients. It's not going to be a cure, but it's going to at least prolong patients' life. In terms of drugs, it's a huge puzzle that you ultimately want to find the key, and if you have that and if you develop a model, then you can actually find the cure. And so we're right now in front of the broken puzzle that we want to put together in order to get this picture. We actually hope that that may happen. We study other diseases where this kind of approach has yielded quite successful candidates for drugs. So we are quite hopeful.